Okay, so welcome back to this next video in which we are discussing receptor tyrosine kinases. Okay, so we're currently in the process of discussing the phosphoinositide-free kinase family of enzymes. Okay, and what they're going to do. So we want to see what phosphoinositides are. So we're currently defining what a phospholipid is. So one of the criteria for to be a phospholipid is that you need a long-chain carboxylic acid within your structure. And the example of a long-chain carboxylic acid I'm currently discussing is stearic acid. Okay, so here's the carboxylic acid group, okay, and then we need 18 carbons in total, so we need 17 more carbons after this first one here, okay. 16 of these are going to be in the form of methylene groups, so I don't want to have to draw 16 methylene groups, so once again I'll put brackets around this single methylene group that I've drawn, and then subscript this 16, okay, and that means copy that methylene group out 16 times. And then right on the end you'll then have a methyl group. Okay, so that is then the structure of stearic acid or octadecanoic acid. Okay, and I'll just circle that in green. So that's sort of like the prototypical example of a long-chain carboxylic acid. A carboxylic acid molecule where you have a really long hydrophobic tail which will not interact well with water. Okay, so this is the lipid component within a phospholipid. Okay, so the long-chain carboxylic acid gives you this part of the name. It's the lipid in the phospholipid. Okay, the other criteria that you have to obey in order to be considered a phospholipid is that you also have to have a phosphate group within your structure. Okay, and this is the phosphate portion, or rather the phospho portion. So let me remind you of the structure of a phosphate group. So a phosphate group consists of a phosphorus atom double bound to an oxygen atom with two alcohol groups coming off the phosphorus atom on either side, like so, and then a single bond to another oxygen which has then acquired an electron via ionic means and therefore has a negative charge. Okay, so this is the structure of a phosphate group. So in order to be a phospholipid, you have to not only have a long-chain carboxylic acid within your structure, but you also have to have a phosphate group. Those are the only two criteria in order to be considered a phospholipid. Okay, so now let's move on to what a phosphoglycerolipid is. Okay, so these are phospholipids, firstly. They're all going to obey those two criteria that we've outlined above, okay, but they're also going to be fundamentally based on a glycerol molecule, which is the glycero in their name. Okay, so all phosphoglycerolipids are going to be based on a molecule called phosphatidic acid. So I'm going to start off by drawing a cartoon of the structure of phosphatidic acid for you. Okay, and then we'll see how we can arrive at a more general phosphoglycerolipid from phosphatidic acid, and then we'll see how phosphatidyl inositol is an example of a phosphoglycerolipid. Okay, right, so let me draw a cartoon then of phosphatidic acid. So, phosphatidic acid is going to have two long-chain carboxylic acids within its structure. So these horizontal lines here, which I'm now colouring in orange, these are going to represent the long-chain carboxylic acids within the phosphatidic acid structure. And these long-chain carboxylic acids are going to be linked to the first and second alcohol groups of a glycerol molecule. Okay, so in green, this vertical line here, this is going to represent glycerol. Okay, now, glycerol is the old name for a molecule that would nowadays be called propane 1,2,3-triol. Okay, so although propane 1,2,3-triol is a much bigger mouthful than glycerol, it's a more useful name because it tells us exactly what this is. It tells us that we're dealing with a 3-carbon molecule, propane, where you have alcohol groups coming off the first, the second, and the third carbons of that molecule. Okay, so that's what glycerol is. Okay, so glycerol is going to form the backbone, if you like, of the phosphatidic acid molecule, you're then going to attach these two long-chain carboxylic acids. And by the way, another name for long-chain carboxylic acids is to call them fatty acids. Okay, so you're going to attach these two fatty acids onto the alcohol groups that come off your first and second carbon of your glycerol molecule. Now the abbreviation for fatty acids is FAs, so these are two FAs, so I'll abbreviate them to that. Okay, right, and the way you're going to attach these long-chain carboxylic acids to the alcohol groups of the glycerol molecule is via esterification reactions. Okay, so let me just show this. So, here is our carboxylic acid group here, and I'll put now an R here to mean an arbitrary tail there. Okay, then you've got an alcohol group that comes off the glycerol molecule. Okay, so this is our alcohol group from the glycerol molecule. This is our extremely hydrophobic tail here in R. Okay, and now what we're going to do is we're going to link this long-chain carboxylic acid to the alcohol group that comes off our glycerol molecule. Okay, and the way we're going to do this is we're going to take the alcohol group off the long-chain carboxylic acids group. Okay, and then we're going to take the hydrogen off the alcohol group of the glycerol molecule, and then we're going to attach the carbon of the carboxylic acid group to the oxygen of the alcohol group, and this link that we've formed here, this will be an ester link. Okay, so that's how we're going to attach these long-chain carboxylic acids to the alcohol groups that come off the first and the second carbons of the glycerol molecule. Okay, right. Then we're also going to attach a phosphate group onto the alcohol group that comes off the third carbon of the glycerol molecule. Okay, and this is going to be via a phosphoester link again. So we've discussed how we can add phosphate groups onto serine, threonine, and tyrosine residues. Okay, and we bind those phosphate groups onto alcohol groups. So we're going to perform a very similar reaction here with the alcohol group of the glycerol molecule. Okay, so if this is our phosphate group here, like so, we're going to bring it up and we're going to attach it to the alcohol group of the third carbon of the glycerol molecule via a phosphoester link. Okay, so we're going to take the alcohol group off the phosphate group. We're going to take the hydrogen off the oxygen of the alcohol group. We're going to bind those two things together to make water. And we're then going to attach the oxygen of the alcohol group of the glycerol molecule to the phosphorus atom of the phosphate group. And of course this link is called a phosphoester link once again. Okay, right, so that then now is the complete structure of phosphatidic acid. You have a glycerol molecule as your backbone, and then of the first two alcohol groups that come off the first and second carbons of the glycerol molecule, you have attached long-chain carboxylic acids via ester links, and then off the alcohol group that comes off the third carbon of the glycerol molecule, you've attached a phosphate group via a phosphoester link. Okay, and that structure is called phosphatidic acid. Now the first thing I want to say is that there is not just one phosphatidic acid molecule, and the reason there is not just one phosphatidic acid molecule is that I have not specified what these long-chain carboxylic acids are. Okay, they could be any old long-chain carboxylic acid. They could be stearic acid, they could be palmitic acid is the other example we've seen, but there are loads of other long-chain carboxylic acids. All they have to be is very, very long, and they have to have a very, very neutral structure and therefore need to be hydrophobic. Okay, so you've got those two long-chain carboxylic acids attached to the alcohol groups that come off the first and the second carbons of the glycerol molecule, and then off the third carbon of the glycerol molecule you've then esterified, well, phosphoesterified a phosphate group onto the alcohol group that comes off there. Okay, and that structure now is phosphatidic acid, but the main thing to stress is that there is not just one phosphatidic acid, because I haven't told you what those long-chain carboxylic acids are. Okay, so because there are many different long-chain carboxylic acids you can have there are many different phosphatidic acid molecules you can therefore create. Okay, right. The second thing I want to explain is what the difference between phosphatidic acid and phosphatidate is. Okay, So people will more often talk about phosphatidate than they will talk about phosphatidic acid. Okay, so what's the difference between phosphatidic acid and phosphatidate? Well, phosphatidic acid is an acid, and phosphatidate is the conjugate base of that acid. Okay, so what do I mean by this? Well, let's firstly explore why phosphatidic acid is an acid. So, notice that even once you have attached this phosphate group onto the alcohol group of the glycerol molecule, you still have this group that I'm now circling in blue intact. Okay, so you still have a phosphorus atom double bound to an oxygen atom with an alcohol group. This is one of those groups that has a similar structure to a carboxylic acid group. Okay, so phosphate groups actually, even once they have been involved in a phosphoester link, they still have one of these carboxylic acid groups, carboxylic acid-like groups within them that is intact. Okay, now what can happen is this can donate its proton away into solution just like a carboxylic acid group would. So, if we think about this alcohol group here, okay, if we draw this bond out in a bit more detail, this single covalent bond that we've got between an oxygen atom and a hydrogen atom here, this will have 2 electrons within it. Okay, one of those electrons will come from the oxygen. So let's say that this one that I'm colouring in red here came from the oxygen, and the other will come from the hydrogen, this one in blue here. Okay, so what can happen is that the hydrogen can fall off this oxygen atom basically, and it can leave the electron that it's put into this single covalent bond here behind with the oxygen. Okay, now what does hydrogen consist of? Okay, well, the main isotope of hydrogen consists of a proton with an electron orbiting it. Okay, like so. So, let's say this is our electron here. Okay, and this is our proton at the centre in green here. So the main isotope of hydrogen, which by the way is called protium, consists only of a single proton at the centre forming the nucleus, with then a single electron around the outside. Okay, there are other isotopes of hydrogen where you don't have no neutrons. So for instance, deuterium then has a single neutron in the nucleus, and then finally there's also tritium, which has 2 neutrons in the nucleus, and you can go further, but those are the main 3 isotopes of hydrogen. However, deuterium and tritium are rare compared to protium. Most hydrogen atoms will be protium, okay, and therefore will have no neutrons in the nucleus. So once the hydrogen leaves its electron behind with the oxygen, all that it will consist of is this single proton. Okay, so what can happen then is these phosphate groups within the phosphatidic acid molecule can donate their proton away into solution, and that's the definition of an acid. An acid is some molecule which is capable of donating its proton away into solution. Okay, so what then will you have left over once the phosphatidic acid has actually donated its proton away into solution? So phosphatidic acid is the name for the molecule when it has that proton here. Once it has lost the proton, the oxygen will now have a negative charge because it's gained that electron from the hydrogen. Okay, and the molecule that is left over is no longer an acid. Okay, because remember, the definition of an acid is a molecule which is capable of donating a proton away into solution. Once you have donated your proton away into solution, you can't donate another one. Okay, once you've got a negative charge on that oxygen and no proton left, then you can't donate a proton anymore. Okay, and this molecule is what's known as phosphatidate. So phosphatidate is the deprotonated version of phosphatidic acid. Okay, and it's what's known as the conjugate base of phosphatidic acid. Okay, and this is a running theme for acids that once they donate their proton away into solution, they are no longer an acid molecule because they can't donate a proton away into solution anymore. What you've got left after that donation is then actually a base, because if we look at the example of our phosphatidate here, we have that negative charge on that oxygen, and that can actually accept a proton out of solution, so protons can come out of solution and bind to the negative charge on that oxygen atom. Okay, so therefore these molecules that are left over after deprotonation, these are actually bases now, because the definition of a base is something which can accept protons out of solution. Okay, and they're called the conjugate bases of the acids. Okay, so once an acid molecule has donated its proton away, you are then left with a molecule called the conjugate base of the acid molecule. And phosphatidic acid is the one with the proton, phosphatidate is then the conjugate base of phosphatidic acid. Okay, so that's phosphatidic acid and phosphatidate. Now, under physiological pH, okay, which is around 7.45, phosphatidic acid molecules, of which you do have some present within the cell membrane, will have lost their protons. Generally, most of them will have donated their protons away into solution, and therefore will be existing in the phosphatidate form, which is why people talk more about phosphatidate than they do about phosphatidic acid. Okay, right, so now let's move over to discussing a more general phosphoglycerolipid. So this is phosphatidic acid, okay? This is a phosphoglycerolipid, but it's kind of the core phosphoglycerolipid. It's the phosphoglycerolipid from which all the other phosphoglycerolipids are derived. Now, firstly, let's just check that it is in fact a phospholipid. Okay, so remember the criteria that you have to obey in order to be considered a phospholipid, where you had to have a long-chain carboxylic acid within your structure. Well, we've got 2 of those, so we've definitely obeyed that criterion. Okay, you also had to have a phosphate group within your structure, and we've certainly got 1 of those as well, so we've obeyed that criterion. Okay, so phosphatidic acid is a phospholipid, and in fact it's more than that, it's a phosphoglycerolipid because it's got the glycerol within its structure as well. Now, all the other phosphoglycerolipids are going to contain phosphatidic acid. They're just going to have an additional structure stuck onto them. Okay, so how are we going to attach this additional structure? Well, it's all coming back to this carboxylic acid-like group here, with the phosphorus atom double bound to an oxygen and then an alcohol group coming off the side. This is capable of binding to other molecules, and therefore you can add on additional groups onto the sides of these phosphatidic acid molecules. So basically, we know that these carboxylic acid-like groups that we've got, one of which we've got here, okay, are capable of forming bonds with alcohol group containing molecules. Okay, so if we were to bring along another molecule which contains an alcohol group here, okay, then what we could do is we could form a phosphoester link between this phosphate group of the phosphatidic acid molecule and this alcohol group containing molecule. And all we'd have to do is take the alcohol group off the phosphate group, okay, take the hydrogen off the alcohol group here, combine those together to make water, and then bind the phosphorus atom of the phosphate group to the oxygen atom of this alcohol group containing molecule, okay, via a phosphoester link, of course. Okay, so we can attach alcohol group containing molecules onto our phosphatidic acid molecule, and this is how we create a more general phosphoglycerolipid, by attaching alcohol group containing molecules onto phosphatidic acid molecules. Okay, this now is the more general phosphoglycerolipid. So, we're now going to look at phosphatidyl inositol, which is an example of one of these more general phosphoglycerolipids, where we have taken a phosphatidic acid molecule and attached on an inositol molecule. Okay, so let's now discuss inositol, which is this molecule that we're going to attach onto our phosphatidic acid molecule in order to create phosphatidyl inositol. Okay, so inositol then. Inositol is the old name for a molecule that would now be called cyclohexane 1,2,3,4,5,6 hexol. Okay, so cyclohexane 1,2,3,4,5,6 hexol. And although cyclohexane 1, 2, 3, 4, 5, 6 hexol is certainly more of a mouthful than inositol. Again, it's a more useful name because we can work out its structure from this. Okay, so cyclohexane, firstly, that tells us that we're dealing with a 6-carbon molecule where the 6 carbons are wrapped around in a loop and all the bonds between the 6 carbons are single bonds. Okay, like so. So 1, 2, 3, 4, 5, 6 carbons. Okay, so we're drawing a skeletal structure here. So remember, in skeletal structures, you don't show carbon atoms. They're implicitly shown by corners. You also don't show hydrogen atoms coming off carbon atoms. They are implicitly shown by where you have missing bonds coming off carbon atoms. You assume that those missing bonds are to hydrogen atoms. Okay, so here is cyclohexane then. Okay, so we've got our 6 carbons shown by these 6 corners, and all of the bonds between the 6 carbons are single bonds. Now that means that every carbon has 2 missing bonds. Now one of those is going to be to a alcohol group, because we've also got this 1, 2, 3, 4, 5, 6 hexol here. Okay, but the other is going to be to a hydrogen atom. So every carbon is going to have both an alcohol group coming off it and a hydrogen coming off it. Now you might say, oh wonderful, how simple. However, there is a subtlety here, a big, well, an important subtlety for understanding purposes. Okay, if you imagine, instead of drawing a 2-dimensional diagram, let's try and draw a 3-dimensional diagram. Let's imagine that our 6-membered carbon ring here is sitting nicely within the plane of the piece of paper. Okay, so each of these carbon atoms is nicely positioned in the plane of the piece of paper. That means that the other 2 bonds that come off each of those carbon atoms, what angles are they going to come off at? Well, one is going to come off, let's look at this one as a specific example. One is going to come off at an angle like that, okay, so it's going to be coming out of the page like so. The other is going to be going into the page away from us at an angle that sort of goes in like that, okay. So, each of these carbons, we are going to have to decide whether we want the alcohol group to come out of the page towards us and the hydrogen to go into the page away from us, or whether we want the hydrogen to come out of the page towards us and the alcohol group to go into the page away from us. Okay, so the subtlety here is that there are multiple optical isomers of inositol, basically. Okay, there are actually 9 different optical isomers of inositol that you can come up with. We are not going to go through all of them, okay, because there is only one that's really important within cells, and the optical isomer of inositol that is important within cells is known as myoinositol. Okay, so if you ever hear someone talking about myoinositol, you now know what it means. It's referring to the optical isomer of inositol that is found within cells. Okay, right, so let me now show you the structure then of myoinositol, i.e. what I'm going to tell you is which direction the alcohol group goes off in, and then I won't show which direction the hydrogen goes off in, because remember, we're drawing a skeletal formula, so we don't show hydrogen atoms coming off carbons, but it will be implicitly obvious which direction the hydrogen goes off, because it will be going in the direction that the alcohol group isn't going. Okay, so we'll start over here. So the alcohol group of this carbon is going to come out of the page towards us, which means, of course, that the hydrogen is going into the page away from us, although I'm not going to show that. Okay, the alcohol group that comes off this carbon here is also going to come out of the page towards us, like so. The alcohol group that comes off this carbon here, it's the same story, it's going to come out of the page towards us, and then the hydrogen will be going into the page away from us. Then it will change, okay, and by the way, these bonds that I'm drawing here where it gets thicker towards the alcohol group, those mean coming out of the page towards us. And the bond that then shows going into the page away from us is shown as a dashed line like this, or at least that's the way I'm going to show it. So this means that this alcohol group now goes into the page away from us. Okay, and the hydrogen therefore will be coming out of the page towards us, but we don't show that. The alcohol group that then comes off this carbon is coming out of the page towards us once again, Okay, and then finally the alcohol group that comes off this carbon is going into the page away from us. So this is the structure of the optical isomer myo-inositol. Now, there is a strict naming system for the carbons within the myo-inositol ring. Okay, so if you have an inositol molecule, your first job is to try and figure out whether it can be superimposed on this picture that I have got here. So you'll have to rotate it around a bit and try and superimpose it on this picture that I've got here. If you can, then the way that you will name up the carbons is as follows. The one that is superimposed with this carbon here will be named carbon number 1. The one that's superimposed with this carbon here, number 2, number 3, number 4, number 5, number 6. Okay, so this is the way of naming up the carbons of an inositol ring. You firstly take your inositol ring, superimpose it on this image, and then the carbons will be labelled according to how they're labelled on this superimposed picture that we've superimposed on. Okay, right. This is the main form of inositol that is within cells. We are now going to attach this form of inositol onto our phosphatidic acid molecule. Okay, and the way we're going to attach it is via this phosphoester link that is going to come from the phosphate group onto an alcohol group. Now, you can take your pick. We've got 6 of the things, but it binds to the one that comes off the first carbon of the myo-inositol ring. Okay, so here is our phosphatidic acid molecule, like so. So I'll colour in the components here. So we've got our phosphate group in vivid purple here. We've then got our glycerol molecule in green here. And then we've got our long chain carboxylic acids attached via ester links in orange here. Okay, then coming off the phosphate group attached via phosphoester links, we've got our inositol ring, which I'll just show as a hexagon like so. Okay, and this structure that we've now created is a phosphoglycerolipid because it's derived from phosphatidic acid by attaching on an alcohol group containing molecule. Okay, and it's called phosphatidyl inositol. Now, this is a running theme for the phosphoglycerolipids. Okay, they all contain phosphatidic acid, and they are all in the naming process prefixed with this prefix phosphatidyl. Okay, so this phosphatidyl prefix, this refers to the fact that it contains phosphatidic acid. So all phosphoglycerolipids start with phosphatidyl. Okay, and then the inositol portion tells you that the alcohol group containing molecule that you've attached on is one of these inositol rings here. Okay, which I'm showing as a blue hexagon. Okay, so this is phosphatidyl inositol. So now we're in a position to talk about what the phosphoinositides are. Okay, so phosphoinositides are now derivatives of phosphatidyl inositol. Okay, so the important thing to understand is that this inositol ring that we've attached onto the phosphatidic acid molecule, it has these other alcohol groups coming off these 5 carbons here. And these other alcohol groups can be phosphorylated. They can have phosphate groups attached onto them and via phosphoester links. Okay, so when you attach phosphate groups onto phosphatidyl inositol, that creates you something called a phosphoinositide. So a phosphoinositide is just a phosphatidyl inositol molecule that has at least one additional phosphate group attached on. So let me give you an example. Okay, so an example would be we could phosphorylate this alcohol group that comes off the 4th carbon of the inositol ring. Okay, that would now be considered a phosphoinositide. The name of this, by the way, is phosphatidyl inositol, and now we just have to label up the fact that it's got a phosphate group coming off that 4th carbon, so we call it phosphatidyl inositol 4-phosphate. Okay, so phosphatidyl inositol 4-phosphate is this phosphatidyl inositol molecule with a phosphate group coming off the 4th carbon of the inositol ring. And for short, phosphatidyl inositol 4-phosphate is often abbreviated to PI for phosphatidyl inositol. So this portion here, this just stands for phosphatidyl inositol here. Okay, then we've got 4, and then 4P for phosphate here. Okay, so the 4P stands for 4-phosphate there. PI4P. Okay, right. That's an example of a phosphoinositide. Another key example that we're going to see, so I'm going to give you the examples of phosphoinositides that are actually going to be extremely important to us, is phosphatidyl inositol 4,5-bisphosphate. Okay, so this is going to be where we have an additional phosphate group coming off the fifth one here. Okay, so if you take phosphatidyl inositol 4-phosphate and add on a phosphate group coming off the fifth carbon here, okay, so the alcohol group of the fifth carbon is now going to get phosphorylated. That will create us phosphatidyl inositol 4,5-bisphosphate. And this is why it's important to understand the optical isomerism problem with inositol, and that the optical isomer of inositol that we are using within cells is this specific myo-inositol. Because if you were just to look at the cartoon that I have drawn you here, you would say, why is that not called phosphatidyl inositol 3,4-bisphosphate? Well, the answer is now plain. When we understand that this is a specific optical isomer of inositol, we understand that the third carbon is different from the fifth carbon, okay? The fifth carbon is sandwiched in between two carbons which have alcohol groups going in opposite directions to the direction in which each alcohol group goes. Whereas the third carbon is in between one carbon whose alcohol group goes in the opposite direction to the direction in which its alcohol group goes, but then it's also next to another carbon which has an alcohol group going in the same direction as its alcohol group. So 3 is different to 5 in this very subtle way. Okay, so that's an important understanding point, and that's why I have brought that up at all, so that you can understand why this is called phosphatidyl inositol 4,5-bisphosphate. Now, similarly, phosphatidyl inositol 4,5-bisphosphate is often abbreviated to PI for phosphatidyl inositol, because of course we do have that once again in our structure. Then it's bracketed 4,5, okay, to tell us which which alcohol groups of the inositol molecule, the phosphate groups, are going to come off, and then we put P2 for phosphate 2 or bisphosphate. Okay, so 4,5 here, and then finally P2 for bisphosphate here. Okay, right, so that's phosphatidyl inositol 4,5-bisphosphate, and it's another example of a phosphoinositide molecule. Okay, right, so I think we're now ready to discuss the PI3 kinase enzymes, okay? But I think we'll take a break and start the discussion of the PI3 kinase enzymes in the next video.